I'm Brad Power, and you probably know this is the prostate cancer lab, where we're learning about a lot of novel tests that can help personalize cancer treatment decisions. And today we have a panel, which is the first time we've done this. We'll see how it goes. I want to first introduce Karin Rodland, who is really the person who introduced me to a number of the rest of you, Mandy and Olink. And I don't know if you introduced me to Kristina, but may have been as well. So that all is thanks to Karin. So she's the network organizer, the panel organizer today. And I'll let each of the panelists introduce themselves just very briefly. So your personal background, your organization and its role. And then what we'll be getting into, if you want to speak to it, is sort of where you are in the life cycle of the evolution of this emerging technology. So those kinds of things. Just a few minutes. Karin, why don't you start, please, and then we'll do Mandy, and then Kristina, and then Michael and Marlon.
That will work. So I am a cancer cell biologist who's been studying signal transduction in cancer for close to 50 years. And I use mass spectrometry and mass spec-based proteomics very heavily.
Not connected to peer.
So I am a joint appointee at Pacific Northwest National Lab, which is a major site of technology development in the field of mass spectrometry. And I also have a joint appointment at Oregon Health Sciences University, which is a leader in precision oncology and smart trials. And I do know a lot of people, and I try to get them involved.
Thank you. Thanks for doing that. And Karin was featured on a previous session we have. I should mention that. Again, we have a recording and a transcript and her slides, I think, from that session as well. So just as a foundation for this conversation. Mandy?
Yeah, hi everybody. I'm Amanda Paulovich at Hutch Cancer Center. I'm a clinically trained medical oncologist with also a PhD in genetics. Unfortunately, I've seen this disease of cancer from many sides. I've been a cancer patient, having been diagnosed with early stage breast cancer at the age of 40, which I guess technically makes me a cancer survivor now, cancer researcher, and a clinical oncologist. As an oncologist treating patients, I was very struck by the variability that I saw from patient to patient, where patients with what looked like the same tumor would have very different responses to the same therapy and very different profiles in terms of side effects as well. And that led me to yearn for what today we now call personalized or precision oncology, which is what we're trying to achieve. I did a postdoc at one of the major genome centers that completed the human genome sequence. That was a very critical start on our path to precision oncology, but it's not enough. Genes don't really that reliably predict response to treatment, and the gene mutations or expression in cells don't reliably predict the expression of proteins in our cancer cells. And since proteins are the targets of therapies, we have to be able to quantify proteins. And when I got into this field 19 years ago, and still today, actually, the vast majority of studies on protein use conventional technologies that are 50 years old and wholly inadequate to meet the needs of the post-genomic community. In fact, there aren't good assays for measuring the vast majority of human proteins in a clinical setting. And this very critical human proteome, as it's called, it remains clinically inaccessible. So in 2003, I joined Fred Hutch to set up a translational proteomic lab to try and leverage new technologies to solve this gap based on mass spec. I'll ask you, Brad, if you want me to describe that a little bit. I can throw up a slide, but I also realize you're watching the time. If that's enough.
No, go ahead. And Mandy, I was going to ask you also to talk about, again, we're very interested in this transition from research to clinical use. And I know you've been associated with something you developed in your lab that's become commercially available recently. So I think that's fair game. And I'd like to hear about that as well, please.
Okay, I'll try to be brief. This is a slide I often use at the end of talks that I give. So, this is sort of the arc of progress, and Karin Rodland actually has been here through most of this, where we've been colleagues in the National Cancer Institute's clinical proteomic consortium called CPTAC. And as I mentioned, in 2003, I came to Fred Hutch, and a couple of years later, we launched this NCI CPTAC program and spent 5 years and probably $30 million convincing the world that these conventional technologies that are 50 years old aren't the only way to measure proteins, that we can use newer technology based on mass spec to make reliable measures of proteins. And that got us another 5 years of the program at NCI, where we spent 5 years and another $30+ million, proving what, in my opinion, was common sense, which is that measuring proteins adds value over just sequencing DNA. And that finally was proved beyond a shadow of a doubt, to the point that even the genomics-centric folks at the National Cancer Institute are now on board, that there's value in combining proteomics and genomics and so-called proteogenomics. And then we spent the last 5 years and another $40 million laying the groundwork for the clinical translation of mass spec-based proteomics. There's a lot of proteogenomic characterization centers looking at the gamut of human tumors, trying to characterize them on a molecular level using mass spec-based technologies. And we also, for the first time, introduced translational research centers whose goals are to try and take these technologies and push them into the clinic. In that phase, we saw the development of laboratory developed tests, CLIA tests. In fact, my laboratory is now a CLIA environment. And we have successfully translated this new mass spec-based measurement technology into clinical trials, where we run patient samples for correlative studies in our CLIA environment. And that's where we're really stuck right now. And in my opinion, this is where the field needs to get. We need to achieve clinical translation, not just in a CLIA environment, but in a regulatory environment, meaning FDA approvals. And we need precedent for that before pharma, before this technology is de-risked enough that pharma will begin to adopt it in its late phase trials, potentially leading to companion diagnostics. Reimbursement for diagnostics in general and for mass spec in specific is woefully inadequate. It's very difficult for laboratories to provide the service and stay afloat. And then new technologies always meet the challenge of physician uptake. And so these are areas that I'm looking strongly at in the next phase of my career. How can we now usher these things a little bit further forward? And Brad, you mentioned the commercialization as well. My lab under this NCI program has developed a large catalog of assays that depend on a valuable and expensive to make reagent called a monoclonal antibody. And we make all of that publicly available through open source portals that the National Cancer Institute maintains. And that's for non-commercial research use. And expert labs can go get and run any of those assays that they want. But to fully democratize access to this new technology, we need to make it available also to laboratories and research groups that don't inherently have those technologies in-house. It's expensive and complex to use mass spectrometry. And so to try and solve that problem, we recently announced a partnership with CellCarta, which is a global contract research organization based out of Canada that does correlative studies for clinical trials, and all of our assays will be made publicly available. They've licensed Fred Hutch's rights to all those assays to make them available to the community as well, to try and get this out there. And we really need to take up these new technologies. And a critical reason is cancer phenotypes and predicting drug response are far more complicated than a single gene mutation or the measurement of a single protein, which is what we typically do now. If we really ever want to achieve precision oncology, we have to figure out, both on the science side and on the institutional side, how to embrace multi-analyte diagnostics that have many proteins, maybe coupled with some genes or gene expression, and patient data. And in the end, this information is going to go into algorithms that are going to output scores that put patients into categories for treatment selection. But our institutions are not set up to deal with any of that. So we now have lots of institutional barriers that have to be broken down. And we need new mass spec technology to enable that part on proteins, because the old ones really just measure usually one protein at a time. So I think I'll stop there, Brad, and happy to answer questions later.
Yeah, thanks very much. I think you set everything up. Like in that arc is everything we hope to touch on and dive into in more detail. That was a great setup. Kristina, why don't you go next?
Sure. Thanks, Brad. Thanks so much for the invitation to be part of this panel. It's a great pleasure to be together with Karin, Mandy, and colleagues from Olink. My name, I'm Kristina Beeler. I'm Chief Business Officer at Biognosys. My key responsibilities at the company are to really transform our technological leadership in mass spectrometry into insightful solutions for our biopharma partners. I lead a global team, a commercial team across Europe and the US. I have scientific background, a PhD in immunology and oncology. And I have been fortunate to see that, especially the rise in the use of proteomics and the entry into the clinical space of this technology, especially in the last several years as part of the Biognosys. Maybe I'll just give you a little bit more information about the company, about Biognosys as an institution. So we have been out there for 14 years, pioneering the field of mass spectrometry. We are co-inventors of a technology called DA, or Data Independent Acquisition Mass Spectrometry, which has truly revolutionized the field of mass spec proteomics, and now has become the method of choice, basically, for large-scale biomarker studies. We have developed a set of proprietary platforms that serve our biopharma partners, from the early R&D, early discovery, to the late clinical trial applications. So we are active across this research pipeline. We have more than 800 customers across the globe that trust our services and products. Headquartered in Zurich, Switzerland, but we have recently opened an office in Cambridge, Massachusetts, and are expanding our US footprint. And that's in a nutshell. I'm very happy to share more.
So I'm in Cambridge, so this is where I get to say this is the biotech center of the world, because we've got so many. We've got San Diego, we've got Seattle, but anyway, I've got to put in my plug for Cambridge. Just one quick question, Kristina. You said most of your customers, or I guess maybe all of your customers, are biopharma. How do you see, and we can dive into this a little bit more, but just a quick answer, how do you see the insights that you're developing, either by identification of biomarkers or maybe predicting drug response or predicting drug resistance? How do those translate? Things that would be useful for biopharma, we could repurpose for clinical use, and how does that translation work?
So that's a very interesting question, Brad, because if we look at the history of the commercial activity of Biognosys as a company, initially in the early days, we were mostly working with the early adopters of the technology. It was GA, was novel technology. People had their questions about the approach. So really the adopters of that technology were the technology savvy people who were excited about novel technologies. The application was in early R&D setting, basic research, small scale studies, basic research questions. And with time, in the last 7-8 years, we've actually seen a push of the market for the technology to be adopted much later down the drug discovery pipeline. So we've seen the adoption of the technology in preclinical setting to understand mechanism of action of drugs, to identify novel drug targets. But also most recently, we have been applying the technology in clinical trials as pharmacodynamic markers. Our clients have been waiting for the data to make decisions on the next cohort enrollment. And this is something new, something that a few years back, we could never imagine that our technology would be used for such decision making. This is still in clinical trial setting, then taking that to the next step, that's still quite a walk to go.
Thank you. Okay, Marlon and Michael of Olink.
Sure. Thanks so much for having us on. We're really excited about the opportunity to be talking with all of you. I'll give a quick introduction of myself, although I won't be the star of the show. Mikhail will be, which I've called Michael forever. So thanks, Brad. But yeah, so I've worked in proteins my entire life. I believe strongly that proteins is where we find a lot of the answers that we're looking for, just like Mandy was speaking of, and the rest of you have said. Our platform, Olink is a platform provider for proteomics, and it's an immunoassay based technology that utilizes the power of antibodies, but then also the power of next-gen sequencing. And so our antibodies are bound to oligos that are complementary, and so it's two of them. They bind to a protein and they hybridize and are elongated and amplified via either qPCR or next-gen sequencing, and it enables us to be able to do up to 3,000 proteins in one sample, in a very small amount of sample. It's something that's been very complementary to a lot of mass spec methods, and it's a very exciting spot to be in. And I'll let Mikhail give himself a little introduction as well.
Marlon, just a quick question, and I should know this, but a recurring issue on this forum is, like, we call it the issue of tissue. And so it's, what is the source material? You said it doesn't need much material. What is the source material? Is it blood? Is it fresh? Is it FFP?
So generally speaking, it's we validated our platform on plasma and serum. However, it has been optimized for many different sample types, including tissues, including cell lysates, supernatants, CSF, aqueous humor, and ocular fluids, urine. We've had pretty much all sample matrices on our platform, and although they aren't validated entirely, they are optimized and have shown to work really well.
Thanks. Mikhail.
Yeah, so I'm Michael Förster. So my scientific background, I actually did a PhD in proteogenomics, like way back at Karolinska Institute, working on basically circulating factors and how genes control, or actually polymorphisms control cytokines in circulation. And then I took the long way around. I did a postdoc, thought, okay, complex diseases is too much. Let's do something simple. Let's do something like immunodeficiencies. And that's how I stumbled on cancer, because I worked on a gene there that was also a factor, a susceptibility factor for hemato-oncology malignancies. And from that, I actually exited the academic world and went into like liquid biopsies. I worked a little bit on liquid biopsies in prostate cancer. So liquid biopsy-based tests for prostate cancer diagnosis, and then later on, kind of like genomics diagnosis for all kinds of cancers. And then very, very recently, I joined Olink, where I oversee most scientific studies in terms of, in one way or the other, that involve like targeted oncology, different solid cancers, hematological cancers as well, and then sees to the implementation of proteomics, always asking what can proteomics add to, kind of like all the questions that genomics cannot answer. So that's kind of like my mandate in a nutshell, and that's why I'm here today.
Thank you. Just one quick follow-up question. We're going to be having Peter Kuhn and his colleague, I think it's Diane Shishido from USC, and he was a founder of Epic Sciences. And so a lot of this, we're going to be approaching this also in a future session from the perspective of liquid biopsies. So Mikhail, if you could just comment for a moment about, liquid biopsies. When you start, we had a previous session, we're talking about spatial, we're talking about single cell, we're talking about proteomics. They seem like four separate topics, liquid biopsy being the fourth, but it seems like they're all the same topic. Maybe it's all integrated. Could you sort of speak to that? So one is sort of the source material, and there's a lot of research and energy into liquid biopsies because of all the advantages they provide. So if you could just speak to that and how it intersects with proteomics.
Yeah, I mean, in terms of like how we see like capture proteomics adding in terms of liquid biopsy, since our library is geared towards serum and plasma, it's kind of like genomics always is the way back time machine. We look at something that's happened way in the past. You know, you had a cell that underwent oncological transformation because of mutations, and you try to pick these up and then kind of like make predictions for therapy in the future. And where we see proteomics growing is way more real time in terms of you can monitor proteins, you can monitor biology real time, you can monitor cancer related changes real time, and then through putting basically the biomarker information you derive way closer to the therapeutic intervention, we think that then you can gear therapies better to specific patients. And if you use capture proteomics or if you use mass spec, this is not an either or. You can, I mean, both technologies have their advantages and have their disadvantages, and should be seen complementary. Then when it comes to clinical implementation, I do think there it's always going to be somewhat, I mean, people will be more familiar with immunoassays. So it's when it comes to regulatory approval, then people might want to switch to an immunoassay based platform, but that's still to be seen. That verdict is still out there. So that's kind of like how we see what proteomics and what capture proteomics can add.
That's really fascinating. I have a background in software, and I see Brian is here as well. He was at HP. And so people who look at long-term industry trends in software, there was a methodology called waterfall, which is a big bang theory of software development. You design, you code, and build, and then you test, and then you implement in big, releases. And then they went to Agile. And Agile was if you do week-long or 2-week sprints, and you're constantly putting more code and more software out in the marketplace and getting feedback from the market. And it seems like that's the same thing you're pointing at. The proteomics is, so if you had genomics, that's hereditary, that's like pretty much locked and loaded, and it's going to be pretty stable over time. Proteomics is more volatile, I guess. It's at the front line. But if you can then have a more continuous monitoring of it through something like blood, you're both getting, it's sort of like the live action versus the blueprint that's somewhere back in a warehouse. It's
absolutely.
Absolutely.
I mean, yeah. And then in terms of like, we always think that proteins kind of like are the best of both worlds in terms of they correlate with what you want to measure, but they don't fluctuate too much that you can, I mean, you basically have way too much noise and way too little signal. So that's kind of like where we think like proteins is at the intersection. It has all the good things of being agile, while at the same thing being stable enough to actually yield a diagnostic test. So.
Okay. Thank you. Let me do one more introduction. And Shino, I know you're here informally. You weren't supposed to be here, but you are. So just if you would introduce yourself and just do a quick comment introducing mProbe.
Sure. Thank you, Brad. So my name is Shino Taiparambal, and I'm the Senior Director of R&D and the site head for mProbe. Some of you might know this company by the name Oncoplex Diagnostics, and then which got bought out by Nant, and eventually now it is in the hands of mProbe. So we are a tissue proteomics company that specifically does laser capture microdissection or laser microdissection of the tumor tissue, and then analyze for 72 different protein biomarkers in a CLIA CAP environment. We've been doing this, we've been CLIA certified since 2013. And we also have a New York State Department of Health license as well, which in fact, the inspection is going today out for the recertification. So over the years, we've been fortunate enough to be in part of the journey for many cancer patients. We've done an analysis on thousands of samples across different cancer types. And in some instances, I was telling our colleague of mine, some instances we get to see those outlier biology that we see, and then we communicate with the oncologist, and then they act on it, and the patient has gone from hospice back to the hospital setting. So those things where you have seen in clinical proteomics in action, that's a moments that we live for. And my training's been in mass spectrometry. I've done my PhD under the NCTR FDA stuff under Dr. Rick Edmondson and group. And then my postdoctoral fellowship was in multiple myeloma. And then I joined this company in 2009, back then expression pathology. And over the years, we worked with some of the key opinion leaders in oncology, been able to do a lot of retrospective clinical trial analysis and demonstrate the power of mass spectrometry in actually predicting clinical outcome. And this is all coming from FFPE tissue proteomics.
Thank you. All right, so we've used about half of our time, and now we've got some open discussion. I could ask questions for the remaining half hour, I'm sure, but I'd like to turn it over to the audience. And if you would raise your hand using that Zoom feature, and I'll call on you, and then you can ask questions of the panel and or any individual that you'd like. Any questions from the audience? Oh, Saeed, please.
Yeah. Hi. Yeah, my main question is related to the reproducibility of this technology. This is the #1 issue with all the data set we are using, we are analyzing, we analyzed at least more than 500 omics data. And the biggest issue with compared to the other omics data, protein data is the least reproducible. Thank you.
Okay, who wants to take that on the panel? Talk about reproducibility. Karin, you want to?
So it depends upon what level of reproducibility you're talking about. If you're talking about technical reproducibility, do I get the same proteins from triplicate aliquots of the same sample? Mass spectrometry is as good as RNA-seq or any other analysis technique. If you are talking about biological replicability, if I take 3 samples of the same cell line grown in 3 different dishes and measure them by proteomics, again, it's comparable to RNA-seq, but because there may be some threshold issues with mass spectrometry and some variability at the low end sensitivity range, maybe it's not quite as good as some other methods, but really the CVs are very comparable between proteomics and RNA-seq, and I'll ask Kristina and Mandy to back me up on that. Where mass spectrometry has gotten a bad rep in your world is that if you look at human studies across populations and you look at DNA mutations, you can replicate DNA mutations across populations, but you don't necessarily replicate biomarkers, things that have been labeled as a biomarker at the protein level. And a lot of that has been due to a couple of factors. The learning curve in experimental design for proteomics experiments over the past 20 years. And I think all of the bad rep that mass spec has gotten has been from the early 2000s, when people didn't really know how to design a serum or plasma proteomic experiment and didn't understand the limitations of mass spectrometry in terms of experimental design. So if you take a look at current studies, like the CP-TAC studies, they have all done a discovery and a confirmatory set where they have looked at proteomics on 100 tumors and then gotten a separate set of 100 new tumors, and every single major biological finding has validated in the confirmatory cohort.
Let me explain this one. You know, say I want to just build a risk model using omics data. All of them, they have a problem of reproducibility. But I can show you. The problem is this one. When we have 1,000 patients, we divide it to the cancer, no cancer. I'm just giving you an example. And then we find the biomarkers. We build a model. Now we want to use this model for new data set. Completely different. The new data set cannot be applied on the old model. And we cannot just mix them together because in the future we have a completely unknown cases. We cannot just mix them and normalize them, right? This is the main problem. We cannot use the same test based on the existing samples for a new set of the patients. That's the core issue we have with this type of data sets.
I do not think that is a true statement anymore. And I'm going to find you in a second a preprint that is just out. I'm going to have to share my screen.
Yeah,
Well,
Karin looks, just to back up some things she said.
Yeah.
I think what you're talking about isn't reproducibility, it's validation of biomarkers in independent patient cohorts. Reproducibility refers to the analytical precision of a platform over replicates, which properly won with standard operating protocols has been extensively shown. That
reproducibility here is based on the polygenic risk model. It means using this model.
That's validation, right? And so all omics problems have that, right? You know this at least as well, if not better than I do, is that when you have a relatively small n compared to the dimensionality of your data, you're gonna find signatures that look like they work in one patient cohort, and many of those don't replicate in another patient cohort. Those are just sort of false signals because of the low sample size relative to the complexity of the signature. That's a different issue from reproducibility, and that issue is not specific to mass spectrometry. That issue is true for transcriptional profiling data as well.
I correct
experiments, scans, right?
It's based on the definition, but this is what's the value of this? There are 5 million experiments. We don't have even 2 biomarkers out of this amount of data. How we can resolve this issue, call it validation on the new data set, that's okay too. Because right now, when we spend $5 million and test on 1,000 cases, they said, we found the biomarkers. Now we go and validate it on the new data set, it doesn't work. Then what's the value? What's the value?
That's no longer true.
It's like credit scoring. I get 10,000 cases, build a credit scoring, give it to the bank, and bank says, "Oh, sorry, it doesn't work." This is a huge issue, actually.
I don't think we can speak specifically to your specific 1,000 case, but you're right in the bigger picture sense that there's been a lot of investment in omic technologies with disappointing output on the other end. And I think that's for a variety of reasons, one of which the technologies, as Karin alluded to, have and people's knowledge of how to use them has had to mature. And I think what Karin is saying now is that that's the case, and I would agree with her. 2 is there's a lot of people doing omic technologies who don't do it very well. They don't have lockdown platforms. They don't have standard operating protocols. The experimental design is woefully inadequate to have the rigor that you need, even with a sample size of 1,000 patients, in order to find a statistically sound signature that has a chance of validating an independent patient set. You need highly annotated samples, you need high quality biospecimens, and you need a very specifically defined clinical question with a rigorously defined phenotype to have a hope, because these are complex phenotypes. These are not simple single gene, single protein phenotypes. And for example, in the last round of CPTAC, we have a 64 protein signature that predicts about 33 to 50% of platinum refractory ovarian cancers upfront at diagnosis. And that signature is robust to validation in 2 independent patient sets. So now we have a multiplexed, or almost have a multiplexed targeted mass spec assay to try and validate that. And we run up against those problems on the left end of my slide. How do we get access to the proper samples and break down these barriers? How do you get reimbursed for stuff like that? How do you get uptake? Because it's a 64 protein panel. It's not measuring HER2 in breast cancer, one protein, or EGFR mutations in lung cancer, one gene mutation. That's never going to get at the complexity of the biology, nor are the sample sizes that we're all stuck using, because the funding agencies have woefully underfunded these studies to have the appropriate statistical power.
Do
So
you
there's a
have
lot
any-
of problems,
do-
none
yeah,
of
do
which
you
are
have any-
specific to proteomics.
Sorry, do you? No. I said all of them. They have a problem. It's not.
I agree.
And do you have any? Do you know any specific experiments which they followed what you mentioned, those rules, which is true? You need to start with a good question and going through that design of experiment. But do you have anything in this field? Do you know any experiments?
Well, I know the work that we've done in this last phase of CPTAC has followed those rules. And
Right.
now the question is, can we keep the project going with funding and samples to get to the larger retrospective validation in the setting of some of these clinical trials? And that comes with its own challenges.
Did you validate it on the independent data set?
3 data sets. It works across the discovery and 2 validation data sets with a similar AUC.
And what was the outcome of that result? It means
What do you
how, what was the predictability power of that?
It picks up with near 100% specificity about between 35 and 50% of the patients with refractory disease.
Sorry, I'm asking technical, but how about the positive predictive value of it? Anything, because sensitivity by itself is not that much. It's a good one, but how about the sensitivity, recall, precision, all those things? Because, for example, Grail has a test, right? Grail has a test, $1,000 test for cancer. He has the 99.5% specificity, but 44% positive predicted value. It means 60% of patients, they label it as a positive is wrong.
Yes, we required that the test be accurate nearly 100% of the time, because for this test to be actionable, you're ultimately potentially denying a patient standard of care. So there's no tolerance for inaccurate calls in this particular test.
Any number for positive predictive value in your test?
So
It's 100% accurate in a subset of those
100%
patients.
accurate, it means there is no error. Absolutely
At the
no error.
threshold, we're probably getting into the weeds here, right? But at the threshold that we set for the ROC curves, it required that there not be false, depending on how you define it, false positive results. But I'm assuming, Brad, this is way more in the weeds than you get in this discussion. And we can take this offline.
Let me, I don't know, Brian hasn't spoken, but I'd like to cue Brian. One of the things we find really useful is to go to the patient perspective, and that's a point of integration. So there's a lot that's going on here, but it comes together at the patient. So I'd just be curious what Brian is listening to in this conversation, what questions he would have.
Yeah, so I have my RNA-seq information. I know, 20,000 genes where I overexpress relative to a pan-cancer cohort of about 12,000 patients and a prostate cancer cohort of about 375. We know that my RNA-seq information is a proxy for the protein expression of my cancer. What is the best way for me to go about integrating proteomics into the definition of my cancer? And is it going to be materially different than the RNA expression data that I have already?
So what question are you asking with the integration? Are you asking the clinician to come up with a personalized therapeutic regimen for yourself? You're asking for prognosis? I mean, those are actually 2 separate questions. You're asking for prognosis, you're asking for personally guided treatment.
It's not prognosis. It's guided treatment. So
Okay.
it could be a personalized approach. Yeah.
If I was doing an N of 1 research experiment on you, I would take your RNA-seq data and the biological processes that have been implicated in the RNA-seq data, and I would use Mandy's targeted assays with her company that she's gone to, and I would verify which kinases in that pathway were actually upregulated and driving the abnormal behavior in the pathway. And I would try to find the nodal kinase, the driving kinase in the pathway by a substrate, phosphorylation substrate analysis. So I'm talking research level stuff, but it would be possible in a clinical trial. And then I would look for small molecule inhibitors of the kinase that I thought from the phosphoproteomic data was driving your disease.
Makes sense.
Okay. Now, and I've been in on several of these discussions. Looking at you as a research project, I know exactly what to do. How do we get somebody who has the clinical power and the funding to look at you as a worthwhile research project and do that very labor intensive, brain intensive analysis of your data? And how do we turn that into the standard of practice for everybody else? Because obviously the people who subscribe to Brad's program are intellectually privileged, if not economically privileged, you know, and you're working hard to access stuff that's beyond the standard of care. So there's two separate parts of the question. How do we deal with you and make it available to you? And right now you're a research subject. You just have to find a physician, a clinical trial that is a precision oncology clinical trial to look at all these different molecular measurements and integrate them and get yourself enrolled. And then the question that Manny is trying to focus on is how do we turn that into standard of care?
Makes sense. What about heterogeneity? So I'm seeing very early right now, I can't verify this, hopefully I'll know next week, but a year ago I had 3 lesions. I was recently scanned, CT scan, PSMA PET scan. It's possible that 2 of the 3 lesions are no longer there, or at least they're not showing up on the scans. So there's something different about my third lesion. So there's heterogeneity. How do we account for heterogeneity using proteomics? Do we use tissue? Is it blood? Is it a combination?
Heterogeneity, you're going to have to do at the tissue level, and that's where the spatial and the single cell comes in. And every single tumor and metastasis is going to be heterogeneous, and every single treatment regime is going to exert a selective pressure. And we don't understand that process well enough. Mandy, please, kick in. You're the MD here. But and that's the reason why we have not yet cured cancer when it becomes metastatic disease, because the evolutionary process in each met is unique. We'd have to sample each met. We don't understand the evolutionary process right now. We have a tendency in clinical trials to give drugs serially because we don't know enough about the combination effects when you use drugs simultaneously. And I have seen people argue that the current sledgehammer approach we use to chemotherapy to try to kill all the sensitive cells simply provides a desert in which the weeds can grow and is actually promoting the outgrowth of the resistant cells. So we may be 10 years from now, we may discover we're doing everything wrong. I don't know. I honestly don't know.
Yeah, I mean, I agree with most of what Karin just said. I mean, we don't generally offer treatment if we don't have data that it improves outcomes. So there may be better ways to do it, but it's not like we're making people worse in general. This this is the issue, right, Brian, that you are kind of at the cutting edge and asking questions that most patients don't ask and that researchers are still grappling with, but it's a really real and imminent issue for you. And Karin's kind of right. You're stuck, as you know, kind of advocating for yourself and finding allies on this journey that are willing to go down this sort of path with you outside of standard of care. And it becomes a bit of a research endeavor. I don't know if you've seen, there's an inspirational story I was just re-looking for online about a patient's, I'll put it in the chat here, describes how a patient's wife saved his life. He had a life-threatening bacterial infection that was resistant to all antibiotics. His organs were shutting down, and she read online about a bacteriophage, a virus that could kill these bacteria. And she had some high-ranking academic position, I think, in the UC system, and so was a well-connected, very smart healthcare consumer, and was able to track down partners in terms of physicians and researchers and rally them around this cause to find a phage that would kill this bacteria that her husband was imminently dying from, and was successful. And you're kind of in that space, right? Because you want the next generation of what's going to be offered in medicine, but that, as Karin said, hasn't become standard of care yet. And you really, you need partners in likely in large academic centers that are willing to kind of go out on the land, and centers that are, legally adventurous enough that they're willing to kind of go down that path with you. Unfortunately, it's unfortunate that it's that hard, but I think, as Karin said, you're pushing the envelope beyond what standard of care is. And I would agree with most of the other stuff that Karin really said.
Well, I'm going to share with you a little bit about my story, which is I did do RNA-seq analysis with Tempus and our other partner, Rick Stanton. And through that analysis, I learned that I overexpress for a few genes, again, relative to these other cohorts. One of them happens to be B7H3 or CD276. And so I had a conversation with my oncologist last September. I said, hey, found this looks interesting. And she was a little bit dismissive about it at first. And then I came back 3 months later. I didn't even bring it up. She said, hey, I want to thank you for bringing that data to me. It turns out there actually is an ADC from Daiichi that targets B7H3. I said, I know. But where I want to go, it's like, okay, great. I have 21 different treatment options that have been provided by this amazing crowdsourced community that we have here. And we've whittled that down to about 8. And then, there's going to be a bit more, back and forth that we go on to 8. But this ADC is definitely in the top 3, if not number 1 right now. And so I look at that story and I say, okay, great. I've had my DNA sequence. It kind of told me a few things. Wasn't terribly compelling in terms of treatments I would use. I did a Pembro using some of the DNA seq. It really just wasn't super helpful. But the next stop in this journey is proteomics. And so I want to I want to show that, like, look, okay, we went to RNA-seq. We found something. What are we going to find if I get into the proteomic area? And what are the treatments that we can identify, and how much better would they be than an ADC that targets B7H3? That's the story. That's my story that I'm sharing with you in preparation for conversations that I'm going to have with the Prostate Cancer Foundation and my oncologist, obviously, and others. I don't know. Is that a compelling story?
Yes. So this is one of the things that the CPTAC program and others is really working on, is integrating the data and using it to do a better predictor. And that's what my PTRC, which was devoted to acute myeloid leukemia, did do. And I'm going to share a screen for a minute. If this works. So can you see this figure here?
Okay.
So this is from a paper. It just showed up on Clinical Proteomics today, and I'm touting my own work. And this is using 3 different bioinformatic methods to predict the response of AML cells to about 25 different small molecule inhibitor drugs. And the higher the bar is, the lower the average error, and therefore the more value there is to that particular type of molecule as a predictor. And you can see in this color here, the proteomics is like the best. Sorry, Sadie. But when you combine the mRNA and the proteomics, which is in this goldenrod color, wow, that's better than mRNA alone, which is in the teal color. So here's RNA alone, and here's RNA plus protein. Here's RNA alone, here's RNA plus protein. The genes here are better than the RNA, but not as good as the protein. So we're developing tools. That's where we are right now. We're at the process of developing tools as to how we combine these types of data to improve the selection of likely therapeutic drugs that you will respond to. And the advantage of this PTRC was that AML is a blood cancer. We have the tumor cells in solution. We have them frozen down in DMSO. We can take them out and plate them into 360 well plates, treat them with dose response curves from 100 or more small molecule inhibitors, some of which are FDA approved, some of which are not. But this gives us actually the density and richness of data that you can use AI methods on and get a valid result. And so one of the limitations of old-fashioned proteomics with Western blots is you could only do, well, let's even say RPPA. You could do 200 things at a time. That's not enough for AI to work on. It's just not. And you were doing 20 patients, 50 patients. So CPTAC is now going to 200 patients. That's still relatively small for what you need to do mathematically, but at least it's approaching the level where things do validate in alternative, in an independent cohort. So now I'm going to stop sharing and shut up and let somebody else say something.
Well,
Thank
I'm
you.
watching the time as the time manager, and I'd like to wrap up by asking each of the panelists to maybe make a comment. We've had a little discussion. I feel like we've only barely scratched the surface. But maybe we'll go in the reverse order that you were introduced. So Mikhail and Marlon, anything you'd like to say?
Yeah, I thought I'd just chime in quickly about specifically like the power that's required for these studies to be actionable. You know, they've been alluding to a large number of samples, and we piggyback off that, and we'll say that that is one of the things that's most detrimental to just one signature. But we do find that proteins are where the action happens, and you know, you're more likely to find something actionable in the protein side than you would anywhere else. And you know, Karin's data definitely showed that.
Mikhail? You're on mute, sorry.
We like not cancer specific, but looking more at like populational cohorts. We recently had more and more studies that use like large proteogenomic screens, kind of like trying to make diagnosis or predictions for common complex diseases. And what will come out, this is not yet out, what will come out, it is a study associated with the UK Biobank, which just released their first big proteogenomic screen. But one of the researchers there specifically asked the question, okay, what can genetic risk scores versus protein risk scores add for all kinds of common complex diseases? And it is actually like that, like proteins. I think in 95% of the cases, proteins were superior than doing genetic risk scores. So we would hope that this is the same, that the same thing holds true for cancers. The question is always, what is the value in terms of like easy access of tissue versus looking at tissue itself. So that's kind of like where where the rubber meets the road, and I think where the future of proteomics lies. Maybe for cancer, it is absolutely be the case that you have to look at the primary tissue, and you won't find a lot of additional information in circulation. But that's still yet to be seen. For other common complex diseases, that seems to be the case. It's the other way around, that actually liquid biopsy-based tests on proteins have a good chance of predicting or giving at least prognosis, and hopefully also predicting response to treatment.
Thank you. Kristina.
And maybe just to recap from my point of view, the key topics that we've discussed today: complex biological processes. They are characterized not by just one level of omics. They need the multi-omics level, and Karin's data beautifully illustrates that. Proteins, I think all of us here in the panel are convinced they provide functional information, essential information to understand the true phenotype, but actually combining all this omics is key. Combining different proteomics technologies is key to understanding complex diseases. But what is also more important is really the reproducibility of the technology, so having robust technologies performed in a well-controlled environment, workflows that have technical reproducibility, but have also the study design are well-powered to bring the answers that we're looking for. And yeah, those are all aspects that we see as part of the success of the studies that we help our commercial partners initiate. These are all key aspects to that.
Thank you. Mandy?
Yeah, hey, I guess it comes back to me of what we really need are better diagnostics, right? What Brian is asking for is a better diagnostic. What I was asking for as a patient and as a doctor treating cancer patients were better diagnostics. But we live in a capitalistic society, and there's not a lot of money in making diagnostics. There's a lot of money in making the next ADC or blockbuster drug, and it's bankrupting everybody. And 80% of healthcare decisions are based on diagnostic tests, but they account for less than 20% of healthcare costs. And if you look at global investment, it's super low. So how do we solve this and overcome that and other institutional barriers? And I don't know the answer to that, but I do think precision oncology is never moving forward until we do, and in any real way beyond what we're kind of already doing. And I've spent months now going around talking to congressional lobbying groups and professional societies, and I don't see anybody out there lobbying for exactly this that we're talking about. And then I've said, well, how about a patient advocacy group? And then what I hear back is, well, those work, but they take 20 years to get anything done, patient advocacy groups. And I don't know if that's true or not. That's the belief out there. So, and I don't know whether this is something you guys think about, but, we have to figure this out. Somehow diagnostics have to be valued in the same way that these blockbuster drugs are. And they're way, way undervalued right now. I just don't know how it feels moving forward beyond where we are, except for tiny little steps that might have little incremental advantages. We need a leap forward. And so I don't know if that's something you guys can think about in this group, but a way to catalyze that would, I think, move it forward for Brian and everybody in that kind of situation.
That's really helpful. It does kind of frame the issue that I need to represent to the Prostate Cancer Foundation and what I need to represent to my oncologist when I see her on Monday. So, I have been on this diagnostic journey, DNA, RNA. Yep. Okay, great. The next step is proteins. And if we're all saying the same thing, Karin just showed incredible data. So it's just like the answer's there. We don't have to get all hung up about N of 1 versus, larger cohorts. Let's just start. Let's start. And you never know what you're going to find when you start. And we have a growing patient network. We have you know, some really, really amazing people that are supporting me. You all are part of that, and I thank you for that. And it's powerful. And so I think I've got a pretty compelling case. I naively believe that, and I won't change otherwise. So, you know, this story is to be continued. I know it's tough. I, you know, spent some time in healthcare. I know how tough it is to move the needle. I'm not naive, but I'm also pretty determined. So this will be continued. And I know where I'm going. And you guys have helped me to, you know, figure out what the objective is. And it's simple. So thank you.
It seems like we should close on that from Brian, because that seems like a complete like eulogy or something. But I do want to give Karin the last word.
No, I mean, Brian has said it all, and I said most of what I wanted to in defense of proteomics right before we started this. I am going to make one statement. This group is focused on patients who already have late-stage cancer, and they're trying to treat their disease and get a better treatment. But wouldn't it be better if we could prevent the disease in the first place, or detect it very, very early when a simple surgery was a complete cure? And that's the other part of liquid biopsies and DNA versus protein. And we're doing all we can to help Brian now that he's in the pickle he's in. But we want to help Brian's son or brother or cousin not ever have to have prostate cancer, because they detect it very early and it's curative. And, you know, Mandy benefited from that in breast cancer, because that field has progressed to the point where early detection is pretty commonplace, and therefore it's treatable.